Metastasis is the leading cause of cancer-related deaths. It consists of a series of steps including tumor expansion, intravasation of cancer cells into the circulation, their survival in the circulation, followed by extravasation and expansion at distant sites. Without further ado, let's focus on the intravasation step. Intravasation is a process whereby cancer cells detach from the tumor mass and penetrate through the endothelial walls of blood vessels to reach the bloodstream, where they're known as CTCs or circulating tumor cells. This step can occur through entry into blood vessels or lymphatic vessels. Next, there are 5 main processes of metastasis, which includes invasion, intravasation, circulation, extravasation, and colonization. However, we are going to focus more on the intravasation process. One specific mechanism involved in intravasation is the epithelial mesenchymal transition, EMT, a complex biological process that endows epithelial cells with mesenchymal characteristics, enhancing their invasive and migratory capabilities. During EMT, cancer cells lose their cell-cell adhesion and acquire a more motile and invasive phenotype. As cancer cells undergo EMT, they gain the ability to breach the basement membrane and invade the surrounding stromal tissue. This process is facilitated by the secretion of matrix metalloproteinases, MMPs, enzymes capable of degrading the extracellular matrix components. MMPs, particularly MMP2 and MMP9, play a crucial role in breaking down barriers, allowing cancer cells to move through the interstitial spaces. Once cancer cells have invaded the surrounding tissue, they approach blood vessels. However, T cells play a role in immune surveillance, which is the body's mechanism for detecting and eliminating abnormal cells, including potentially cancerous ones. T cells can recognize and target cancer cells for destruction. At the same time, interaction with endothelial cells is facilitated by various adhesion molecules and receptor-ligand interactions. Cancer cells can exploit these interactions to adhere to the endothelial lining, eventually crossing the vessel wall to enter the bloodstream. Besides, tumor-secreted factors can influence the behavior of endothelial cell lining blood vessels. This may also include promoting the expression of adhesion molecules on endothelial cells, facilitating the attachment of cancer cells into the bloodstream. The successful completion of these steps signifies the accomplishment of intravasation, marking a pivotal point in the metastatic process as cancer cells embark on their journey through the circulatory system to establish secondary tumors in distant organs. The intravasation process is regulated by intrinsic, microenvironmental, and mechanical factors. For intrinsic properties of cancer cells, it refers to characteristics or features inherent within the cancer cell itself that contribute to its growth, survival, and dissemination. These include genetic mutations, epigenetic changes, and gene expression that give cells an intravasation advantage. Genetic mutations. First of all, the behavior of cancer cells can be greatly influenced by gene mutations, which may make them more likely to intravasate into blood vessels during the process of metastasis. So, mutations can alter immune response and cause microenvironment modifications. Interactions with the immune system can be affected by gene mutations within cancer cells. By avoiding immune surveillance, mutations can result in altered immunological checkpoints or decreased detection by immune cells, which can facilitate intravasation. Gene mutations also can change cellular connections and signaling within the tumor microenvironment. Alterations in the expression of certain genes in the tumor microenvironment could facilitate intravasation by providing a favorable environment for cancer cells to spread out and penetrate blood vessels. Epigenetic alterations. Afterwards, epigenetic alterations that can influence gene expression patterns and cellular behaviors in several ways, which may trigger cancer cells to intravasate. The activation of epithelial mesenchymal transition, EMT, related genes by epigenetic changes can provide cancer cells with mesenchymal characteristics and increase their motility and invasiveness. Due to its ability to allow cells to separate from the main tumor and invade neighboring tissues, this transition is essential for intravasation. The expression of genes involved in cell adhesion and motility also can be affected by epigenetic changes, which may affect how cancer cells interact with the endothelial cells that line blood vessels. Variations in the expression of adhesion molecules affect how cancer cells adhere to the walls of vessels, which promotes intravasation. Gene expression. Also, gene expression profiles can predispose cancer cells to intravasate into blood vessels during the process of intravasation, through a series of molecular alterations that enhance their invasive potential and promote their interaction with the vascular system. So, gene expression will influence upregulation of invasion-related genes and changes in cell adhesion molecules. Upregulation of invasion-related genes or overexpression of certain genes linked to invasion, like those regulating matrix metalloprotein MMPs, promote the extracellular matrix's breakdown, which permits cancer cells to enter surrounding tissues and possibly even blood vessels. Then, changes in cell adhesion molecules refers to changes in the expression of molecules that promote cell adhesion, such as integrins, affecting the attachment and interaction of cancer cells with the endothelial cells that line blood vessels. This could encourage the adherence of cancer cells to the walls of the vessels, also allowing for intravasation. Metabolism of cancer cells. Uneven metabolism in tumor cells may affect tumor growth due to their metabolic adaptability and heterogeneity. They can rewire their metabolic patterns for survival, proliferation, and invasion. For example, the primary tumor tissue may impact metabolic processes due to variations in organ vascularization and oxygen availability. Glycolysis. One of the key players in cancer cell survival is glucose metabolism. Serves as the main energy source. A byproduct of glycolysis, methylglyoxal, YAP-mediated metastasis may be able to trigger spread through. In addition, excessive dependence on glycolysis leads to the buildup of lactate. The generation of lactate may contribute to a shift in pH, which in turn might trigger the activation of cathepsins and MMP9. These enzymes are responsible for breaking down the extracellular matrix, ECM, and facilitating invasion. Mitochondrial metabolism. Oxidative phosphorylation still plays a significant role, though the extent of this process may be variable in different cancer types. Oxidative phosphorylation was enhanced in cancer cell lines when glycolysis was inhibited. Oxidative stress occurs when there is an imbalance between the production and detoxification of reactive oxygen species, ROS, which are often formed as byproducts of oxygen metabolism. ROS has been acknowledged as a factor that promotes the spread of cancer cells to other parts of the body. It achieves this by either causing damage to DNA or by activating signaling pathways that support the spread of cancer cells. Hypoxia and tumor vascularization also are key features involved in cancer progression. High amounts of hypoxia master regulators HIF-1 alpha and HIF-2 alpha are linked to worse patient outcomes and more cancer spreading to other parts of the body. Another study suggested hypoxia may induce intravasation by activating CXCR4 signaling, leading to the adherence of cancer cells to endothelial cells. Fatty acid uptake metastasis is also affected by fatty acid metabolism. Mutations in free fatty acid absorption routes linked with invasiveness. Fatty acids may trigger oncogenic signaling via incorporation into membrane structure or oxidation. Cells in the microenvironment. There are many types of cells in the tumor microenvironment, such as immune cells, endothelial cells, active fibroblasts, and parasites. Their modes of operation involve modifying the characteristics of cancer cells through signaling pathways, changing the tumor microenvironment, or directly participating in the process of intravasation. Fibroblasts exist in many organs, and when cancer grows, they can be activated or recruited to the tumor site. This is why they are called cancer-associated fibroblasts, CAFs. This type of cells has been linked to different features of tumor growth, such as tumor growth, extracellular matrix, ECM, remodeling, angiogenesis, invasiveness, and metastasis. Neutrophils are innate immune system cells that are found in large numbers inside the growing tumor. Neutrophils can indeed facilitate the spread of cancer to other parts of the body, particularly by establishing a specialized environment in distant organs known as a pre-metastatic niche. Neutrophils can promote the spread of cancer cells by releasing structures called neutrophil extracellular traps, nets. Macrophages are the cells of the innate immune system, and when recruited to the cancer site, they are referred to as tumor-associated macrophages, TAMs. Invasive tumor cells polarize and move towards blood arteries via a paracrine loop involving macrophages. Tumor cells respond to macrophage-secreted EGF, and macrophages respond to tumor cell-secreted CSF1. T cells. T lymphocytes are adaptive immune cells that often get into the tumor surroundings as the disease gets worse. It has been found that CD4 plus T lymphocytes help with increased intravasation by either directly signaling to cancer cells or indirectly by controlling TAMs. Platelets have been linked to the growth of new blood vessels and the spread of cancer. Parasites have been considered as gatekeepers of metastasis. Indeed, the extent of parasite coverage and their combined effect might be different depending on the type of tumor, where it is located, and the stage of tumor development. Adipocytes are the primary components of adipose tissue, responsible for both fat storage and the secretion of lipid and protein molecules. Furthermore, they are the primary cellular constituent of breast tissue and become activated during the development of cancerous tumors, therefore transforming into cancer-associated adipocytes. Adipocytes can potentially facilitate the movement and entry of cells by modifying collagen structure.